Welcome back. This is the second video on muscle contraction, and in this part we're going to learn about the phases involved in muscle contraction and relaxation. So basically there's going to be 4 major phases, and then once we understand all this we'll go through the entire process. The first phase is what's called the excitation phase. And in this part, what happens is those action potentials which are traveling down the axon to the axon terminal to the terminal bouton and that nerve fiber are going to, through their neurotransmitters, cause a new action potential to occur in the muscle fiber at the neuromuscular junction. The second phase is what's called excitation contraction coupling. There are going to be some events that are going to be linking the new action potential on the sarcolemma of that muscle fiber to getting those myofilaments ready to contract. The third phase is the actual contraction phase, where that muscle fiber develops tension and hopefully will shorten. The thing we talked about previously called the sliding filament theory. We're going to go through it again in detail. And the fourth phase is going to be the relaxation phase, where now we no longer have any stimulation, so the muscle can return to its resting length, and will be ready to be excited again. So the first part of muscle fiber excitation, here is our setup, our neuromuscular junction. So we're going to have a nerve impulse, our action potential, traveling down the axon to the axon terminal, arriving in the terminal bouton. Who remembers what happens next? When it arrives there, that impulse is going to open calcium channels. Now these are not ligand gated because there's no neurotransmitter that has to bind to them. So these are what are called voltage gated calcium channels. It's the change in the voltage which opens these calcium channels. So when that impulse travels down to the voltage gated calcium channel, it's going to open, causing the influx of calcium ions, which then is going to make those synaptic vesicles filled with their neurotransmitters undergo exocytosis, releasing their contents into the synaptic cleft. Okay, so what happens now to those empty vesicles? Well, they're not going to stay right there at the surface. They're going to back away from the surface, and they're going to let other ones that are filled have the primo spots in case a new nerve impulse comes down so they can release their neurotransmitters. Okay. And so those empty ones which have backed away can now be refilled, because maybe they weren't completely empty. Maybe they were only partially emptied. Okay, so we left off with acetylcholine being released in the synaptic cleft. And what it's doing is it's looking for its receptor on the postsynaptic cell. So in this first part, we had one nerve impulse or one action potential, which traveled down the axon, hit a voltage gated calcium channel, causing the influx of calcium, which caused synaptic vesicles filled with neurotransmitters to undergo exocytosis. It actually causes around 60 synaptic vesicles to undergo exocytosis, and each one of those releases about 10,000 acetylcholine. So when I say they're releasing their neurotransmitters into the synaptic cleft, where the one neurotransmitter we're thinking about, acetylcholine, is looking for its receptor on that postsynaptic cell. All right, here is what happens next. That acetylcholine is going to bind to its ligand-gated ion channel, the cholinergic receptor. 2 of them are going to bind to them. Now notice on the motor end plate. Okay. So that one neurotransmitter is looking for its ligand-gated receptor on the motor end plate where it's going to bind. When two of them have bound to it, that ligand-gated receptor is going to open its channel. Sodium is going to start pouring into the muscle cell, and much slower potassium will start exiting the muscle cell. And so this is going to give us depolarization in the muscle cell, and now that voltage change gives us an action potential in that postsynaptic muscle cell, which now is going to spread out in all directions. Okay, on the pictures we're going to have an arrow showing it going in one direction, but I want you to realize it's not just one direction. It's going to spread out in all directions. So here's a closer look, showing acetylcholine being released from the synaptic vesicle of the terminal butone. So acetylcholine are the red dots. This is the motor end plate on the sarcoplasmic reticulum. You can tell because it has these infoldings, and the green structures are the cholinergic receptors. So two acetylcholines are binding. Do you see two of them binding here? That's why this one's open. This one only has one bound, so it's not open. So two have bound here, lots of sodium entered. The red line with the squiggle is showing the action potential being produced, okay? After that, what's going to happen is the acetylcholine gets kicked off. It's here in the synaptic cleft where acetylcholinesterase is located, and so it'll be broken down into the acetate, which can be, which it can diffuse away, and the choline molecule, which will be reuptaken into the terminal butone. All right. So now the impulse is spreading along the sarcolemma as represented by the blue arrow over here. So in addition to those ligand-gated cholinergic receptors, where sodium could enter and potassium can exit, we also have sodium gates where only sodium can enter and potassium gates where only potassium can exit. Both of those gates are voltage gated. So as this action potential diffuses away from where it originated, the change in voltage is going to be causing the opening of these voltage gated ion channels on the sarcolemma away from the motor end plate. And so now the action potential travels along the sarcolemma and is going to go down a T-tubule to the triad area where we have the terminal cisterns of the sarcoplasmic reticulum with all the stored calcium right next to it. When it arrives at the terminal cistern, those voltage-gated ion channels in the sarcolemma here are going to be linked to calcium channels in the terminal cistern. So when the voltage-gated ion channel is allowing that impulse to occur in the skeletal muscle, that will trigger the calcium channels in the sarcoplasmic reticulum to release their calcium, as seen in the top of this picture, or the bottom of this picture, I guess it is. So the channel's open, calcium diffuses into the sarcoplasm. It's going to diffuse away from the side. It's going to find troponin binding sites. It's going to bind with the troponin, change the shape of the troponin, pull the tropomyosin off the binding sites inside the actin molecule, let the little myosin heads enter and form the cross bridge. Okay, so let's talk about myosin just for a second. Here's our myosin molecule with this one showing two heads on it, and I want to point out this green area is the binding site where it's going to go into the actin, and that's how it forms the cross bridge. In addition to that, there's two other binding sites. One contains ATPase, which is the enzyme that will break down ATP to ADP plus inorganic phosphate. So in order to initiate contraction, this myosin head must have ATP bound to it, which is the third binding site. So ATP comes and binds. The enzyme in the myosin head hydrolyzes the ATP. And now what we have is we have a myosin head that has an ADP and inorganic phosphate. And what that does, I realize you can't see me, is it cocks the myosin head. I don't know how to, okay. So if this is the myosin, basically it puts the head going up at an angle, okay? So it's ready to contract. All right, so this is the high energy form. Okay. So once it has bound to the binding site in the actin, that releases the inorganic phosphate from the myosin head, and that gives us the power stroke, which basically means this cocked myosin does this, okay? It's going to pivot. Here's where the M line is, the center of the sarcomere. It's going to pivot towards the center of the sarcomere, basically ratcheting the actins closer to the center of the sarcomere. Now, it gets to ratchet one time, and then it's stuck because it requires a new ATP to bind to it before it can relax. When the new ATP binds to it, that actually destabilizes the cross bridge, which is what breaks the cross bridge, okay? And that gives us the recovery stroke, where now the enzyme in the myosin head breaks down that new ATP into ADP and PI, and it's recocked, ready to go. So when the myosin movement is that power stroke and then the recovery stroke, okay? So it's not that it has to do things twice, it's doing this part and then that part. And then it's ready to do it again, okay? Because it is stuck in this position until there's a new ATP. When there's a new ATP, it recocks and it's ready to go again. It will not require another ATP. All right. So I want you to stop it on this side. Start up here at 10 o'clock where it says start here and talk yourself all the way through this process and just keep on going again and again because that's what it's going to be doing. All right, with that, I want to thank you. Make sure you understand this part before we continue on. I will see you shortly.